Hello and welcome dear students. In our today's lecture, we will be discussing about identification, procedure and methods. In this lecture, we will highlight various methods of identification and correct determination of plant names. Identification in biology is the process of assigning a name to an individual based on its body features such as morphological, anatomical, physiological, behavioral or molecular. The term determination may also be occasionally used as synonym for identification. The process of plant identification, whether scientific or traditional, has been an age-old activity of human beings and depends upon the criteria and the system used for the purpose. It is a process to determine the similarity or differences between two organisms. That is, two organisms may be same or they may be different. It implies the comparison of certain characteristics of the unknown plant with the known plants, or simply the process of matching specimens to a known taxon and determining its correct name. We know that a plant species is a single kind of plants, or more technically a group of actually or potentially interbreeding plants. A genus is a group of related species, a family a group of related genera, and order a group of related families, and so on. Collectively, all of the major plant groups or divisions constitute the plant kingdom. This is how the living world is classified. Once a system of classification has been set up, it becomes possible to identify an unknown plant by comparing it with the members of the groups that have already been recognized. According to Blackwelder, identification enables us to retrieve the appropriate facts from the system to be associated with some specimens at hand, and is better described at the recovery side of the taxonomy. Now, we discuss various methods of plant identification. The most traditional means of learning the identity, function and use of plants is through teachers, family and acquaintances who live within the same environment. While this is less common today, it was the norm for thousands of years. As a matter of comfort and survival, children would learn from their parents and other elders about all the plants surrounding them. Individuals who exhibited a particular knack for plant use and identification would evolve into herbalists and healers. These people still exist in many places and often are happy to share their knowledge of plants with interested students. Learning about plants in this way provides a depth and connection that are difficult to acquire through book learning. Some of the common methods of plant identification are as: number first is expert determination and recognition. Then we have comparison with correctly named materials in herbarium. We take the help of taxonomic literature and field guides. We utilize keys and similar devices, and we also take the help of internet. Now, we discuss these methods one by one. The first is expert determination and recognition. It is the most reliable and accurate method of identification, provided the time and availability of the expert is not a constraint. For an expert to identify a specific group of plants, he or she is supposed to have available floras, monographs and revisions and other relevant literature for that specific group. The expert recognition and identification reliability is based on extensive past experience and knowledge of the expert identifier with the plant group in question. In some groups, this is virtually impossible. Now, we come to next method, that is comparison with correctly named materials in a herbarium. The second most important method of identification is the comparison of unknown with correctly named specimens, photographs, illustrations or descriptions. However, this method is workable only when there is sufficient material for identification in the form of reliable named specimens in the herbarium or museum, photographs, illustrations, or descriptions. A herbarium is a collection of pressed and dried plants mounted on standard sized sheets bearing a label arranged according to a sequence and available for reference or study. The herbarium comparison is most easy and generally more reliable method, as the recognized herbaria are generally housing specimens identified by experts and the specimens are revised from time to time. Majority of the herbaria in the world have a wide ranging collection of specimens and offer facilities for on-site identification or having the specimens sent to the herbarium identified by experts. Researchers can personally identify their collections by comparison with the duly identified herbarium specimens. Now we discuss about taxonomic literature and field guides in plant identification. Various forms of literature which incorporate descriptions, illustrations and identification keys are important resources for correct identification of unknown plants. Important forms of literature which help in the process of plant identification include floras, manuals and field guides, monographs and revisions, illustrations, journals, abstracts and indexes, etc. Flora is the inventory of plants of a definite geographical region, whereas a manual represents somewhat exhaustive treatment including descriptions, keys for identification, ecological data, status, chromosome number and glossary of the particular group of plants of a region. A guide is a compilation of limited amount of information, but generally includes information like list of plants, identification keys, descriptions and illustrations, which are very helpful in plant identification. Botany students often carry field guides on hikes and journeys to help them identify unfamiliar plants. Some field guides use flowchart techniques while others are less formal. Generally, field guides will group plants together based on similar characteristics. Then offer one or more pictures of the plant along with a description of its habitat and characteristics. Plants, particularly trees, frequently are portrayed in winter and in summer as an aid to identify them with and without leaves. A monograph is a comprehensive study of a particular taxon, such as a family or a genus, etc. It has exhaustive information of the taxon, including literature reviews, nomenclature reevaluation, and information from all other possible sources gathered and analyzed. It helps to produce a clear taxonomic treatment of a taxon, often stabilizing the classification. A revision is a less comprehensive treatment and includes studies of the specific group within a taxon, generally carried out to resolve taxonomic confusions in that group. Now we come to fourth method of plant identification, that is use of keys and similar devices. The use of keys or similar devices, that synopses, outlines, etc., is by far the most widely used method and does not require much time, materials or experience involved in comparison and recognition. Keys are most direct way of identifying plants. A botanical key is a series of specially arranged statements working through a key separates plants with and without key characteristics and gradually narrows down the number of possibilities. Eventually, if all goes well, the number of possibilities is reduced to 1 and the specimen is identified. Various types of keys and key formats are used in plant identification, such as single access sequential or dichotomous keys, which include indented keys and bracketed keys, also numbered keys, and multi-access or polyclaves, which include body punctured cards, edge punctured cards, and tabular keys, and also interactive keys, which are computer-based multi-access keys. The single access or dichotomous keys have been most popularly used devices in identification of plants from more than 200 years. These keys work by offering a fixed sequence of identification steps, each with multiple alternatives, the choice of which determines the next step. If each step has only two alternatives, the key is said to be dichotomous, else it is polychotomous. Multi-access or interactive keys allow the user to freely choose the identification steps and their order. Now, we discuss something about the procedure for identification using dichotomous keys. A dichotomous key is a device for identifying unknown organisms to some taxonomic level, that is up to species, genus or family level. It is constructed of a series of couplets, each consisting of two statements describing characteristics of particular organism or group of organisms. Each statement of a couplet is called a lead. A choice between the two leads is made that best fits the organism in question. The leads typically begin with broad characteristics and become narrower as more choices are required. To determine an unknown plant with the help of a key, read both leads of a couplet before making a choice. Even though the first lead may seem to describe the unknown material, the second lead may be even more appropriate. Having compared our plant with the leads and decided which lead fits our plant, we then need to be shown which couplet we must proceed to next. Both these needs are met in keys. However, they differ in different typographical formats. In a bracketed key, each couplet has its two leads immediately adjacent under the same left hand number. The first couplet to be consulted stands at the hands of the key next to the number 1. The next appropriate couplet to consult is indicated by a reference number such as 2, 3, 7, etc. as shown to further down the key. Place it on the right hand side of the chosen lead. These numbers direct the user to the next appropriate couplet to be consulted. In case of indented key, each couplet has its two leads indented by a same amount from the left hand edge of the page. The first couplet to be consulted is the one least indented and which has its first lead at the hand of the key. The next appropriate couplet to be consulted is the one with its first lead immediately below the chosen lead of the previous couplet, its leads being the next least indented pair below the latter as depicted below. Now, we discuss about interactive keys. An interactive key is an interactive computer program in which the user enters attributes, that is character state values of the specimen. The program eliminates taxa whose attributes do not match those of the specimen. This process is continued until only one taxon remains. Computer access keys involves the principle of multi-access keys, that is polyclaves, rather than sequential or single access, that is bracketed keys, and they may not be greatly affected if only one or two of the many characters do not fit the sequence. Now, we discuss about computer and internet aided identification methods. Internet is an ideal way to find information about uncommon or difficult to identify plants. Because of the extent of information available, it serves as a data bank for vast number of species. Websites ranging from amateur and enthusiastic sites to professional botanists and university departments. It is relatively easy, given the proper computer software, to convert multi-access keys to a computer interactive format. There are many interactive keys and databases available online which are nowadays utilized to a great extent for quick and correct identification of unknown plants. Some of the major databases and internet based interactive keys are listed as interactive keys of Flora of Nepal, PlantNet, that is Flora Online interactive identification key, or Delta, that is descriptive language for taxonomy, a descriptive database from which descriptions, a variety of identification aids and analysis of relationships can all be derived. Then we have Mika Pandora, that is a database program for plant identification. And we have International Plant Names Index, that is IPNI, which is a database of names and associated basic bibliographical details of all seed plants. IPNI is the product of collaboration between the Royal Botanical Garden Kew, the Harvard University Herbarium and the Australian National Herbarium. We also have Index Herbariorum, a joint project of the International Association of Plant Taxonomy, that is IAPT, and the New York Botanical Garden. Is a detailed directory of about 3,000 plus public herbaria of the world and about 8,800 staff members associated with them. We also have Kew records of taxonomic literature. This valuable resource lists references to all publications relating to taxonomy of the flowering plants, like gymnosperms and ferns, along with reference to phytogeography, nomenclature, chemotaxonomy, molecular taxonomy, chromosome surveys, palynology. All these are also included along with the relevant bibliographies. The database currently contains some 175,000 references published from 1971 to 2000, with the new references being added once a week. We also have Vascular Tropicos, or VAST, a nomenclatural database. This site provides access to the Missouri Botanical Gardens Vascular Tropicos nomenclatural database and associated authority files. Dear students, today we thrashed out about the process of plant identification, its procedure and various methods applied to correctly identify and determine the exact name of a taxon. Hope that it will help you to know about the plant identification and application of various methods for that purpose. Thank you very much. Have a good day.